Okay, welcome everyone. Thanks for being here early morning. So we're here for the PhD presentation. Bryan, who's a PhD student in integrated biology. My name is Patrick Lynn here. I'm Bryan's advisor. Bryan has a co-advisor in microbiology, Martin Kuster, but he is in Zoom meeting. A little bit about Bryan. So not a linear trajectory that many of us experience. So initially, Bryan was on his way to become a famous chef. Has a Gordon Blues. Is that a certificate or title or?
It's a degree.
Okay, so this is back in Minneapolis a while ago, and then went to community college there. Took some math classes, and then eventually moved to Boston, where he got this University of Massachusetts in Boston, where he got a bachelor's degree in mathematics there. Then he came here to work on a specific problem in bacterial populations, where we look at the evolution of cheating. So cheating is the issue that's going on. We test this in a chemostat. That's what Martin Schuster has. Martin is a microbiologist who works with chemostats. So for those of you who don't know, the chemostats are bioreactors. They're kind of vehicles to test, how populations behave. And so I remember Bryan coming here and thinking he would do mostly modeling. And the first time we kind of suggested to him, why don't you do both modeling and experimental work? He was kind of looking in a shocked way at us. So, but I think after some arm twisting and convincing, he embraced it, and I think he has done some great work doing both modeling as well as model analysis, and as well as testing of certain hypotheses. One of the main things is what's called the tragedy of the commons. So, which is a famous paradigm in biology. I'm sure Bryan will say more about it, but one of the things Martin actually initiated this was to ask, can we prove that thing happens, that this phenomenon occurs in a chemostat model? And so initially, before Bryan came, we had established that yes, it was possible, but oddly enough, nobody had actually tested it. And so this is what Bryan really did. That I think is going to be his claim to fame, is to really show that the tragedy also happens in not just as a paradigm, but in real life testing with accurate models, etc. So some of this work is published. I think there's 3 papers published already. A 4th one is in preparation. A little bit of what's coming next. He will be here for 1 more term as an instructor in IB, I believe. And then he will be off to Irvine, UC Irvine, to do a postdoc starting this summer, I believe. So, yeah, right.
Great. Well, thank you for that. And thanks everyone for being here. It's exciting to see faces in the audience. So just to jump right in, I am going to talk about the last 6 years of my life in 40 minutes and tell you all about the chemostat. So first, before I start talking about cooperation, I want to define it. And so for the sake of this talk, cooperation is just going to be an act which imposes a fitness cost on the individual, but then benefits the recipient. And since I have family in the audience, fitness is just a measurement of how well a species survives and reproduces. And there are lots of ways organisms can cooperate. So one example is contributing to a common goal. You see this with something like pack hunting. They can share resources like food. Vultures share their meals with each other, and they can cooperate by refraining from competing. This happens with plant competition for light. But microbes also do all these same behaviors that we think of only really occurring with complicated organisms. Now, resources that are shared amongst populations are also referred to as public goods in evolutionary biology. And my research is going to focus on the responsible use of these shared resources. As an example for this shared resource problem, I'm going to use sustainable fishing. The idea is that each individual is going to be incentivized to fish as much as possible. Each fisher is going to want to catch a lot of fish to sell it, maximize their profits. But as that occurs and more fish get taken from the pool and the population can't sustain itself, can't replace itself as fast as it's being depleted, you get a resource collapse. And we see this with fishing actually in the 90s with Atlantic cod. And so this is called a tragedy of the commons, when that common or public good gets depleted. Now, if the public good is necessary for a species survival, so if there was a population that needed that cod in order to survive, then that population can also collapse. And that is the phenomenon called tragedy of the commons. There's lots of mechanisms that have been identified which help maintain cooperation and therefore avoid this population collapse. These include things like kin selection. So this occurs with eusocial insects that are haplodiploid, which increase their relatedness. This occurs with reciprocity. So this is kind of a tit for tat trade. I'll help you if you help me, trade legs for a voice perhaps. And we see this with vampire bats that will reciprocate their blood meals with each other, but only with other cooperative bats. And this occurs with enforcement. So reef fish use different kinds of enforcement on their cleaner fish, such as punishment or partner choice. These same complicated ways of enforcing cooperation also exist in microbes. So microbes can use biofilms, which are an extracellular matrix of goo that limits their dispersal to increase relatedness. They use signals through a process called quorum sensing that I'll describe in a little bit as a way of reciprocity, and they can use the production of things like toxins as punishment for enforcement. And then I examine the impact of cooperation on the population dynamics in very specific bacteria. So this is the socially cooperative Pseudomonas aeruginosa in a chemostat. Now, Pseudomonas aeruginosa produces several extracellular public goods. This includes siderophores, which are used for iron acquisition, bringing iron from the environment to the cell. They produce enzymes that can break down larger protein in the environment into digestible amino acid products. You could think of that as like a knife that cuts up your steak to eat. And they produce these toxic compounds as well, such as thiocyanin or cyanide. And the production of these public goods is primarily controlled through a process called quorum sensing. The loss gene system is one of the primary genes controlling quorum sensing. The loss I, being the inducer, is going to produce a signal molecule into the environment. And when the cell density is low, so too then is the signal molecule density. But as the cell density increases, that signal density also increases. And once that density is high enough, the cell will then receive that signal molecule by the LOSR gene into its receptor site, subsequently turning on gene expression. So you can think of this as a power strip where the receiving of that signal molecule basically turns on the power strip, and everything plugged into it then also becomes turned on. And so that includes the production of extracellular public goods, such as the enzyme. So quorum sensing requires a sufficiently dense population before the production of these metabolically costly cooperative behaviors can be started. Now, knocking out this LOSR gene makes it so that it can't receive that signal molecule, and subsequently it won't produce any of these public goods. So cells in which we've knocked out this LOSR gene become our social cheaters. They're not contributing to the cooperative behavior of producing these extracellular public goods. If we have a cell that is producing public goods, and in this case, we're looking at the enzyme, that enzyme is then going to bind or catalyze with the substrate, producing these amino acid products that then get consumed for growth and replication. When we introduce these cheaters that don't have the ability to produce the public good, they also then save the cost of production and get to apply that instead to growth and reproduction. And so they're actually going to grow at a faster rate. And this increase in frequency that these social cheaters will exhibit eventually makes it so that there's more cheaters in the population than cooperators, and the population cannot be sustained, creating a tragedy of the commons. And I look at that effect inside of the chemostat. So this is a cartoon of a chemostat here, where the bacteria are grown in the center together. But on the left, we see that the food source, the nutrients, get pumped into the system. So this means that our bacteria are never nutrient limited. And there's an outflow of the entire mixed solution. And so this natural turnover of nutrients is supposed to emulate some of the natural turnover of nutrients you would see in larger ecosystems, as well as the outflow of the mixed solution emulates perhaps the death and immigration you might see in those more complicated ecosystems. Chemostats are also accurately described with ordinary differential equations. So it stays at a constant volume and it's a well-mixed solution. And this is an example of a very simple system of equations that describe a chemostat model. I'm going to have equations up throughout the talk, and I put them up here not because I expect you to walk away having a grand understanding of some complicated math, but I really want to highlight the biological processes that were included in my models. So in this case, we have that flow rate. We have a positive product of that dilution with the nutrient entering the system, and then a negative product of that dilution with everything else in the system being flushed out. And then we have the bacteria consuming and growing from the nutrients that are available. I also think chemostat models have a neat application to larger ecological systems. And I'm not the only one that thinks so. So here we have the gut as an example of a chemostat model. You can see the flow being through the digestive tract and the mixed solution occurring in the stomach. We also see it being applied to river systems. So as the river flows through a certain space. And I say this not because I want to over extrapolate my results, but because I think there's some really cool potential for these chemostat models to have ecological applications outside of a lab. And I specifically used experimentation, classic mathematical analysis, and computational simulations to answer questions about cooperation and the evolution in a chemostat. Using the chemostat as a study system offers the ability to develop these things in conjunction with one another. So I was able to let my experiments inform my models inform my experiments, which isn't always possible when they're done separately. And so my major aims were to look at whether or not the tragedy of the commons does occur in experimental chemostat co-cultures like the theory predicts, to look at evolution, what happens as those populations evolve over time in the chemostat when cheaters are present, and then also to take a look at a few mechanisms of cheater control. So starting with this first aim, this model gets a little bit more complicated than the first one I showed, but a lot of the same processes are there. So we have that same flow rate, that dilution. We have a positive nutrient entering the system and an outflow of everything in there. In this case, we are expanding the nutrient information to include the enzyme and substrate catalysis that's occurring. So we have that rate there turning our larger protein pieces into smaller amino acid products. We have the growth from those products, and then we have the production of the enzyme by the cooperators, and which reduces their growth as well. And so this model predicts that there will be a stable population when no cheaters are present, that the tragedy of commons will occur when there's a mixed co-culture, and that the cheater frequency will increase over time throughout cultivation. My data of my chemostat experiment for the first one, looking at just when no cheaters are present, this is the cell density of two chemostat cultures over time. First thing to note is that our time doesn't start at 0, it starts at -32, and that's because we need to grow the bacteria into a certain densities when they are hormone sensing is turned on before we turn that flow rate on, otherwise we'll flush the system. So at the point of 0 is when we actually turn the flow and the chemostat on. And we see that over time, we do get a fairly stable population over the course of about 8 days. So that first prediction does hold true. Then we want to look at when we add the cheaters into the population. And so starting with a 10% cheater frequency, we're going to have the same thing where we grow it up without that flow on. And then once we turn it on, we see the population collapses. So in this case, the presence of the social cheaters did indeed cause a population collapse. In this case, we have 4 experimental replicates. So that one also is holding up. Then looking at cheater frequency, I want to talk about how I measure cheater frequency. So talk about how our cheaters are, we have this loss-R gene knockout. In addition to that, we had an antibiotic resistance insertion. I should say ABR isn't the actual gene name, it's just my shorthand for antibiotic resistant. But we also had that inserted in there. And so we were able to use an antibiotic plate, trimethoprim, to identify which cells are our cheaters and which ones against our cooperators. So here I have a plate, one of our antibiotic plates. You can see the cooperator, where our cooperator control would be nothing is growing because it doesn't have that same resistance, whereas the cheater has some growth there, and so it has that resistance. We later had some intriguing results that made us add an additional way to measure our social cheater, which is getting into the phenotype. And so in this case, we're looking at enzyme production, and this enzyme that breaks down larger protein will also break down the larger protein in milk. So a very expensive version of carnation powdered milk goes into these plates. And again, you can see with my controls, our cooperator, there's this dark halo around it. That's because the milk has been digested and essentially removed some of the protein there. And then where our cheater is, it doesn't have that same degradation. And so we also looked at whether or not the enzyme was present through these milk plates. With the results, in this case, I've separated my 4 co-cultures into individual graphs. The gray is from the same previous graph showing their cell density over time. But in this case, we got a nice mix of results, the kind of thing that turns a quick first chapter into a quick first book. And so for the 2 on the left, we do see this increase in cheater frequency. Both ways we measure cheater frequency over time. These are my favorite replicates. On the top right, we saw the opposite occur, and this is the one that got us initially thinking to measure, start measuring things a little differently as well. But then on the bottom right, we see one of each, even more intriguing. And so when we think about what defines a cheater, if we define it as not producing the enzyme, and we then focus on that lighter tomato color, then for 3 out of the 4, we do see that increase over time. So we do mostly see an increasing in cheater frequency throughout cultivation, except for that one replicate that keeps me up at night. Okay, so then we want to say, well, how does the model's predictions compare with our experimental data? And so simulating that, we need to find a lot of biological parameters to represent all the different aspects, parameters in our model to fit it. And once we do that, we get a decent fit here with everything, with the population level data. But once you look at the cheater frequency data, there is a bit of a gap there, which I will dig into in I don't know, 3 minutes. But until then, we can also use the model to explore and learn about the system. And so I put this up here not to overwhelm you with an absurd number of figures, but to illustrate that different parameters within the system, different biological parameters have different effects. So some don't really affect the system much at all, and others can have like a pretty large one. But one that I find particularly interesting is our cost of public good production, and that it has a minimum threshold, even without cheaters, otherwise a population will collapse. And so naively, you would think like reducing a factor that limits growth would just always be bad, but that you would want to limit the amount of growth reduction would always increase their growth or the population size, but without enough of a investment in the public good, with or without cheaters, it causes a collapse. And so from this chapter, this experimental side, we do indeed see that the tragedy of the commons occurs in the experimental co-cultures, and that cheater frequency was a bit variable and can't be encaptured by a single deterministic model. The model itself fits well with our data, which is when parameterized with biological values, which is exciting. This really underscores that it has some predictive capabilities and that it can isolate the effects of individual mechanisms and parameters that can be hard to interpret with only experimental data. So moving on to how the population evolves over time. So I pointed out that we had a little bit of a gap in our prediction, as well as some variability within our data. So we want to ask if we can then use these models to predict origins of cheater frequency discrepancies and some of the variability that we're witnessing. So we initially tried and looked at several different possible ways that these cells may have mutated. And as is tradition in science, I'm going to share with you only the one that worked. But looking at our model, we can modify it. And we're thinking, in this case, we are including the idea of faster growing mutations. And so this is, there's a PSDR mutation that's common and creates faster growth. So this was inspired by that study. And so I have an additional equation here for our new mutant that's appearing. What's currently highlighted is what I've already highlighted in previous slides. And so then what we're adding here is we have that this mutation is going to be growing faster than the other cells that are in there. It also has that outflow, and then it's going to have a rate of mutation. In this case, we're looking at a mutation that has a parent of the cheater. So this would mean that it's keeping that antibiotic resistant marker. And when we do that, add this into the model then much better reflects the data for those two replicants. Looking at the version where we have a divergence in our antibiotic resistant frequencies and our protease, our enzyme production frequencies, we then assume that may have been a cheater that evolved from the cooperator parent that wouldn't have had that antibiotic resistance. So we just move the mutation origin to the cooperator into the mutant. And when we do that, we can get that same kind of split effect in these simulations. And then for the last one, where everything went down, in this case, we're looking at perhaps a faster growing cooperator. So still the rate of mutation comes from that cooperator parent, but now we have to add in the cost of enzyme production and the enzyme then that mutant is going to be producing into the system. And we can get a fleshing out of the social cheaters in that case over time. So we can show with some math, but can we actually find them in the chemostat? And so as a reminder, I had these two ways of screening with their antibiotic resistance and that milk production. And so we can then use those screening methods with cells from our chemostat samples. So the top here for the defined are our initial cooperator and cheater. Our initial cooperator having enzyme production, so the milk plates would create a halo, and our initial loss, our cheater, would have that antibiotic resistance, so it would grow on our antibiotic plates. So a cheater from loss our parent is going to actually have the same phenotype in that case of those two plates as our original loss our cheater. Cheater with a wild type parent will have a unique one where it won't express either. And then a faster growing cooperator from the original cooperator parent, the wild type is our way of just kind of saying the original parent. It will have an enzyme production and no antibiotic resistance. And so I can then go to the samples and screen for these two sets of phenotypes. And when I do that and then grow them in a co-culture the same way as I did before to assess their growth, I can compare the relative fitness, which is if it's over one, it means one is growing faster than the other, to the original loss R. So our loss R cheater has a relative fitness against a cooperator as about 1.4. So it grows about 1.4 times faster. You can think of it that way. And then our samples from the chemostat were way higher than that, which is kind of cool. Close to 3 for some of them, with the exception of this one in the middle, but this is one where we can't actually distinguish it from our lost our cheater, so we suspect we just got one of the original guys in our samples. But we got some pretty good, significantly faster growth here to validate that we have these faster growing cheaters in the mix. Okay, and so from this, we see that the models can be used to explore some of these roots of experimental outcomes, and then can inspire experimental directions, potentially saving some time and supplies. If I hadn't necessarily landed on faster growing mutants, I may not have known to do that particular experiment. And I could have spent, who knows, another 3 years here trying to resolve some of this stuff. And then as far as the experiment went, the chemostat selects for faster growing cheaters over long-term cultivation, at least in 3 of our replicants. So the results of these 2 pieces was just accepted into +1, which is exciting. Yay, yeah. And then moving on to mechanisms for cheater control. In this case, we looked at 2 different mechanisms. One is punishment via like a toxic compound production. And the other one is environmental engineering, changing their space using biofilm information. Biofilms are essentially they create this like extracellular matrix along the glass of the wall, almost creating a second niche space for them to live in. So starting with the cyanide production, why cyanide production? It is quorum sensing control, and it is a punishment mechanism that has been shown in other studies to be able to constrain cheater growth and lead to coexistence. So as far as the model goes, we now add 2 equations, which I've tried to mark with little symbols here. We have a punisher and the toxin, and the way that the punisher and the toxin work is much akin to the way the cooperator and the enzyme work, where it's going to produce the toxin at a metabolic cost to itself. And then the toxin itself, affects anyone that it isn't producing it. So it's called pleiotropy when genes are linked. So the idea is that the resistance to the toxin is linked to the production of it. And this model, this chapter was primarily a mathematical analysis. So I'll just be showing some of the simulations and save you from the proofs. But when we have just the cooperators alone, we get a stable population. In this case, our time starts at 0. So if we compare it to the previous data, it would be starting on that end of it, which is great that it is aligning with other results. And when we mix in the social cheater, we get a tragedy of the commons. So that matches some of the previous models that we used in their predictions. When we have just the cooperator and the punishers mixed together, they don't always coexist. So under certain circumstances, if the cost of producing the enzyme is more expensive than the cost of producing that toxin, it actually will still be unstable. Whereas if the opposite is true, it'll be stable. And this is because that punisher is almost also, in a way, a bit of a cheater. It's not producing any enzyme, it's fulfilling a different role, so it's more division of labor. But that enzyme is necessary for the population. So there's a balance happening where there needs to be more enzyme being produced than toxin in the environment. And then when all three are mixed together, we can get mixed results. We can either have a tragedy of the commons, and this occurs if the toxicity effect on the cheaters is too low, or we can get competitive exclusion of the cheater. And this occurs when the toxicity on cheaters is sufficiently high. Essentially, it's just that the effect of the toxin needs to harm the cheater a little bit more than the cooperator, as that cooperator is balancing its total fitness of reducing its fitness from both the toxin and the enzyme production. The results of this study are also published, so that's exciting. And then moving on to biofilms, looking at whether or not biofilm formation can stop a population collapse. So why biofilms? These are also shown to be quorum sensing control. They add spatial structure to the environment, which is something that is shown in cooperation to help stabilize populations. And there is some empirical evidence that they promote cooperation and constrain cheater growth. More importantly, they were really hard to get rid of experimentally. So I also had to throw out a few experimental runs, just because if the biofilm grows too much within the chemostat, it can throw off the dynamics of the system. And so if you can't beat them, join them. I figure if it's so hard to get rid of, then it must be an important feature of the dynamics that are at play and worth including into the model. So this model is even more complicated, but the idea is it's much similar to the first model, but our cooperators and their enzymes just now can reside in 2 locations. So they can both be kind of free floating in the hemostat, or they can be adhered into the biofilm on the wall. So we have new equations for the biofilm as a location within the chemostat. The biofilm, though, isn't infinitely large, so there's some constraints on space that need to be included into the model. And then we have a rate of adhesion for the bacteria, as well as the rate of sloughing off, kind of just being knocked off the wall from the turbidity of the fluid that also gets included. And much like with the previous models, we get that when there's only the cooperators on their own, we get no biofilm stability, and then tragedy of the commons when we have a cooperator and the cheater together. But when we have the biofilm added in, we are able to see stable coexistence. And this is pretty exciting for me, at least, because up until now, most of the models only were able to show competitive exclusion and not stable coexistence. But we can also get stable coexistence as cycles, as limit cycles. And although the amplitude of those cycles and the exact parameters of those cycles varies based on lots of other parameter values, it could have some ecological ramifications as they tend to occur when the number of usable niche spaces increases. So when the fluid state of the chemostat is unstable, but the biofilm is stable. As you transition into a fully stable state, we get cycles. So it's possible that a shift in number of occupiable spaces could lead to limit cycles. So to summarize, these toxin production could theoretically be used as a mechanism to remove cheaters from the population, but with biofilms, those actually, which reduces that diffusion, allows for coexistence. And so now I will summarize 5.5 years in 2 slides. We have that the tragedy of the commons does indeed occur in experimental chemostat co-cultures, just like our theory predicts, that punishment can result in competitive exclusion, the removal of those cheaters, and that this privatization of the enzyme by holding it into that biofilm can actually result in stable coexistence. And then a little bit more methodologically, concurrent theoretical and empirical investigation allowed us to have one inform the other. And so we were able to use our math models to provide some mechanistic insights to guide experimental directions. The experimental data was able to help us identify things like the minimum complexity we need in our model to let it, so that it would still describe our data and ecological outcomes. And then also that experimental observations can inspire new models, as with the biofilm one. And so with that, I'm going to jump into some thank yous. So first, we always have to thank money, but first, more importantly, my advisors, I'm really grateful to both of them, Patrick and Martin. I know I was a atypical choice of a grad student, I think, for both of them, and I hope that they don't regret that. But I also feel really lucky that everyone on my committee, I really took something important and special away from them. So Rishali was a great example of both not just how to do great science, but also how to prioritize the people that do science as well. And Ben, I always really appreciated your enthusiasm with my projects, because I certainly lost my enthusiasm many times throughout the last 5 years. And even my GCR, Eduardo, I think I really appreciated that you inspired me never to lose authenticity in my presentations and gave me permission to include drag queens in them. So thank you for that. The IB staff deserves, I think, put as much work into this dissertation as I did. There's no words that can express my gratitude to them. For my lab, so I feel really grateful to have a bunch of lab mates that are as cool people as they are scientists. I think Parker deserves a special shout out. We were just off by 1 year and we went through the whole COVID thing together, and I kind of asked for like a kinder, cooler, more generous lab mate to deal with having to teach me how to use a lab like an undergrad, even though I'm a PhD candidate. But then I also had like the super lab here in IB. So there's a cool group of people from all different labs over the years. And that really helped me still feel connected to IB, even though my advisors were kind of disparately across campus. Big shout out to the Bi2X folks, especially Carmen and Lori and Adam, who I think really exemplify kind of backseat leadership. The number of things they let me run full steam ahead with is probably too many, but I still appreciate them a lot for that. And then it is a tradition to spend the last couple of slides thanking family and friends. So I'll start with my family, who are big and silly, to no one's surprise, probably. And this doesn't even cover a portion of them, but special big thank you to, I'll say, my aunt and uncle who are here, and their kids, Dan, Joe, and Sarah, who are kind of like second set of siblings growing up. in the way where you still like your siblings at the age of 30 something. So it's a good thing. And then, also my family, just the kind I'm not related to. I'm super grateful for my friends that I had here throughout my time in grad school. I'm just gonna read exactly what I wrote in my dissertation, which is from the BGPPFCD to the salt marsh. Thank you for the joy you brought to an otherwise tumultuous pandemic and broken arm-filled experience that sometimes felt like getting hit by a bus. And if you know, you know. Almost the end, my nephew, who wants to be a scientist, and he is also very good at enthusiastically reminding me that science is actually cool and exciting. And then last but not least, last but best, Pancake, who gives me the best edits every time she walks across the keyboard. She had an unwavering commitment to sitting in my lap during my thesis while I wrote it, and she was great at screaming at me if I tried to stay up late working. So I think I like to think she brings joy to my students and colleagues as well, but mostly to me. So writing the thesis wasn't so bad with her in my lap. And then that's it. Questions?
Bryan. Yeah, so that includes, well, not yet, this questions, right? So, any questions from the audience? Started.
So on Zoom, we have can a living bacterium change from non-cheater to cheater or vice versa? Or non-punisher to punisher, or are they both born with it?
So the loss are deletion is something that occurs as like a natural mutation you could expect in cultures. And so you could go from cooperator to cheater. That one we see a lot. Could even switch back. I'm gonna say anything is possible. Follow your dreams. Yeah.
Yeah, I'm interested in the effect of the biofilm and there's a lot, but the effect of the biofilm on the production of the enzyme, production of the toxin, sort of, are there diffusion effects? So can you maybe expand on that a little bit?
Yeah, the main effect is a diffusion effect. And there's some studies that show this, that essentially the extracellular like polymeric substance matrix that is formed that creates the biofilm just stops the enzyme from like moving away. So they're able to capture some of it and keep it close. And so that's
it's benefiting the wild type,
yeah,
the producers or
yeah, cooperators, whatever you want to call them. Exactly.
And so the cheaters are not biofilm formers.
Yeah, so this is depending on what article you read, is the answer to this one. It's one of those sorts of things where I think it might really depend on exact experimental conditions. There's some papers seem to say that there can be like a patchy-ish effect of growth in biofilms, but some show that the biofilm formation is really extrinsically linked to this quorum sensing circuitry. And so if you're not quorum sensing, then you're not really able to produce those substances either.
So it might be worth pointing out that in this, at least in the model, right, that's so far really all you've done. I mean, no experimental verification yet, is that the cheater doesn't go into the life. So it's like a refuge really for the whole player.
At least with modeling, it's like usually try to start with the simpler version of the thing and then start adding. So I'm the book of ideas that we all have sitting aside that hasn't been touched. There's of course like, oh, you could get into maybe creating those patches, or you could get into what if I add them in and they can take this much space, but we got the simple first edition.
The,
Beautiful research, thank you. You back in your first chapter, they all started at 10% frequency cheaters. And I guess that kind of the likelihood of a tragedy commons outcome might depend on that initial frequency. I'm curious, because like if it's born anew, very low frequency drift will probably take it away. Is there like a range of starting frequencies where you expect a tragedy of the commons to result?
Yeah. So we kind of designed our experiment to hope to create the tragedy of the commons. So we chose a higher frequency to try to support that. It's possible that at such a low frequency, any mechanisms they have that would deter cheaters might exist to deter them from coming up from low frequency. Our initial dream was to do experiments at a range of frequencies. Some dreams die. When we ended up getting some like interesting cheater frequency results, that ended up taking over some of the other experimental stuff we dug into. And we lost the thread of doing like several more chemostats at different starting frequencies. But yeah, I think it's possible that if we started them sufficiently low that we might not see the tragedy. It's totally possible.
Your data showed that you had kind of equilibrium between different phenotypes and genotypes, right? So my question is it possible that these things are all genetically related? Can that be a starting point for diversifying selection? In other words, can they begin to accumulate mutations and diverge and contribute to the really high diversity of natural populations that we know is out there?
Yeah, definitely. I think one of the things we found, right, where there's all these mutations that maybe occurred, we found these faster growing ones, but to imagine that that's the only mutation that occurred in like an 8-day bacterial culture, like they had so much time to do weird stuff in there. I think the expectation would be that ending population is probably quite different than the starting one, especially with the selection pressure of the cheaters being present. It is Lynn, a newer grad student. It's now her project to find out what mutations were all in there. It's my baton of a gift I'm passing.
yeah, there was no sequencing done, right?
But it will be done.
By you or by?
No, by Lynn. Yeah.
All right, good luck. Yeah.
Back in culture 3, is it possible that there are some mechanisms that were triggered in the wild type at the start of 2030 that killed off the cheaters? Like, can you theorize on why that one killed off the cheaters or what?
Yeah. So I would suspect that is probably the case where the mutation occurred earlier. Something potentially even during like the batch phase of it, because it stays this like super closed system to keep it sterile, because a tiny bit of dust will ruin the whole thing that's happened before. And with that one, it's like you kind of expect a mutation from the parent, the cooperator, because they start at a higher density. So just by like probabilities, we would expect mutations from that group, that population over mutations from the cheater. But when I was playing with the numbers to try to see if the simulations could match the data, I do remember with that one, the mutation needed to happen sooner to get the same results.
I guess kind of continuation of that, what's like a typical timeline for a chemostat experiment from like prepping to running and what the data from?
The typical timeline? Typical timeline for the chemostat, including, so before I even like put bacteria in it, there's a bit of a process. So I talked about the biofilms being a pain. we have to treat the glass with basically Rain-X and like bake that on there so that hopefully any drops like fall back into liquid and don't form on the glass. Plus you have to like kind of flush and clean all the tubing. And then when you run the experiment, including the batch phase, about 8 days must take time. So it would be about 3 weeks usually, which isn't as bad as losing a field season. So I try not to complain too bad, but still hurts. Yeah.
So I know your model organism is it's like notorious for infecting people with people suffering from CF lung.
Yeah.
Where like your lung has like pretty much extra nutrients for bacterial growth. And I know it's a thing that like usually isolates from those infections are last R mutants are deficient. How do you think your model can, if at all, describe that phenomenon?
Yeah, so growing on a lung won't have that same kind of like dilution effect, right? So I think there's some interesting potential there with the way we maybe treat wounds with these infections. If you would expect, say, this loss of immune occurs on its own in the lung to then like cause a population collapse, what we need is for these results to occur, it needs to be well mixed, and we need dilution. So could additional wound cleaning then put the results more towards what I saw, maybe?
Okay, well, thank you for the attendance, general public, you're excused now.